Thank you very much. First of all, I'd like to thank the Saudi Society of Blood and Marrow Transplantation for giving me the opportunity to speak to you all today, to the respected Chair Hugo, as well as all the audience. Many of you may already be in a post-perennial mode. So, part of my talk is going to be clinical, partly it's going to be conceptual, and what I'm going to try to do is share with you what's to come, because this is a very exciting time to be a myeloma translational researcher and an early phase clinical trialist. So, you've seen a rendition of this slide in my colleague's presentation, the lunch presentation that highlighted daytime relapse myeloma. But the bottom line, the reason why I'm showing you this slide is, for the most part, we focused on developing drugs based on both normal and malignant plasma cell biology over the past 15 years. So the proteasome inhibitors, the immunomodulatory drugs, for the most part, have been working more directly towards killing the myeloma cell. But we're entering an era where thinking beyond that. How do we modulate the immune system? How do we modulate the bone marrow microenvironment and bring about change in the outcomes for our patients in that fashion? Having said that, from the time when I was a trainee to now, we've seen a fourfold increase in the survival outcomes for myeloma patients. So we're in a good place right now, but things are going to get better. The holy grail of cancer medicine has been how to utilize the immune system to go after cancer cells. I don't want to scare anyone in the room, but 1 in 3 out of people sitting here will develop a cancer in our lifetimes. Our immune system is charged with identifying those cancer cells and taking them out. What happens, the reason why cancer develops in certain individuals is the cancer cells achieve a critical mass where they're informing the immune system not to do their job right. So the usual Treg cells and the cytotoxic CD8 positive cells that are supposed to help kill the cancer cells are no longer in play because of the increase in the cancer cell mass. And what the hope of immunotherapy is to reverse that balance. So what you want to achieve with your immunotherapy is what you see on the left side of this figure, that you include an immunotherapeutic approach in cancer therapy and allow the immune system to start doing its job one more time. Within myeloma cells, just like, myeloma is actually very unique because the myeloma cells grow inside the bone marrow where there is a rich immune milieu along with other players in the bone marrow microenvironment. So how does myeloma evade the immune system? It can do that by suppressing dendritic cells and T cell activation and proliferation by cytokine production. It can downregulate the MHC class 1 on the surface of the myeloma cells so that it prevents antigen presentation to the immune system. It upregulates surface inhibitory ligands such as PD-1, PD-L1 pathway. I'm going to share a couple of slides in regards to that. And it can also recruit counter-regulatory and immunosuppressive cells such as Tregs and MDSCs. So there are many ways in which we can potentially modulate the immune system to make things uncomfortable for the myeloma cell as we introduce anti-plasma cell-specific therapies. So the PD-1, PD-L1 pathway has been exploited ad nauseum now in many different malignancies. We've seen the most data with solid tumors as well as certain B-cell malignancies. Within multiple myeloma, PD-1 inhibitors have actually shown to increase T cells. PD-1 is increased in T cells of myeloma patients, both in the relapse setting as well as newly diagnosed setting. And there's actually a trend of increased expression as patients go from smoldering to active disease. And if you block the PD-1, the survival of mice within the mouse models, the PD-L1 positive myeloma cells goes down. So this makes a rationale to combine an immunomodulatory drug and a PD-L1 specific strategy. Now, what we've seen in the early phase trials, and this is just one example of what I'm showing you, that there is certainly activity in patients who are len refractory You would expect pomalidomide and dexamethasone to give you a response in 3 out of 10 patients, whereas when you combine it with a drug like pembrolizumab, the response rate goes up to 60%. Now, we've seen some negative data from the two frontline, one pivotal trial in the frontline setting and in the relapse setting with Lendex and Pomdex. I was the first author on the frontline study, and you should be seeing those papers and more details about them in the New England Journal of Medicine in a couple of months. The bottom line I'm trying to share with you is there is activity. This is a very potent regimen, and there is more to come. This story is not dead in myeloma. Not just pembrolizumab, but there are other PD-1, PD-L1 inhibitors that are being combined with non-IMiD-based therapies. So this story is not dead. The immuno-oncology angle of modulating the immune checkpoints is going to be moving forward. You're going to be hearing about this in the future. There are trials that are combining different checkpoint inhibitors with other monoclonal antibodies like daratumumab that you'll be hearing about, as well as some of the other novel checkpoint inhibitors. So going from immunomodulation that way to how we can engineer the patient's T cell to go after the cancer cells, there's a lot of excitement about chimeric antigen receptor T cells. And the way that the CARs or chimeric antigen receptor T cells function is by taking the patient's own T cells. You are trying to transduce a chimeric antigen receptor which can identify a cancer-specific antigen, and then you are also activating that T cell before you give it back to the patient. So these kind of CAR T cells, there are 2 products that are already approved for different indications in the United States. I'm not sure what the status of those products is, in terms of availability in Saudi Arabia. But even though ALL and non-Hodgkin's lymphoma have been the first indications, studies are moving very quickly in myeloma. Now, B-cell maturation antigen has shown to be a promising target in multiple myeloma. It's perhaps one of the more specific antigens that is selectively expressed on plasma cells in humans and not on other cell types. And its presence is restricted primarily to the plasma cells and the B cells. So there have been several products that have been designed to target BCMA within the chimeric antigen receptor space. The 2 specific products that I'm going to share with you are the Bluebird BB2121 Anti-BCMA CAR T and the LCAR product, which was developed by Nanjing BioLegend, which has now been taken over by Janssen. If you look at the BB2121 data that was presented at ASCO earlier this year by my colleague, Dr. Nupur Raje, this was on a dose expansion cohort of patients having received at least 150 million CD4 cells. If you look at the median prior lines of treatment, this was a fairly heavily pretreated patient population. All the patients had a prior autologous stem cell transplant. They've had 7 prior lines of treatment. And if you can go down the list on this slide on the right side, you will see that these were patients, a third of the patients were actually refractory to pomalidomide, lenalidomide, bortezomib, carfilzomib, as well as to daratumumab. And 71% had been exposed to those drugs. So generally well tolerated. The cytokine release syndrome was seen in 71% of the patients, but if you look at grade 3 or higher, it was only in about 10% of the patients. None of the patients developed grade 3 neurotoxicity symptoms. The deaths on the study were primarily related to disease progression on lower doses of CAR T cells, and the 2 patients who died were at active doses in complete response. One had cardiac arrest and the other developed therapy-related MDS from previous autologous stem cell transplantation. So this is a very encouraging swimmer plot showing you that over time, patients continue to get deepening of responses. Overall response rate was 94%. 56% of the patients achieving a complete response, and the median progression-free survival in this patient population was almost 12 months. If you look at the carfilzomib single-agent data, daratumumab, pomalidomide, any of those drugs, you will not find this kind of depth of response or duration of response. And these patients were already refractory or exposed to those therapies already. Now, this is the second product that is a little different, targets BCMA, but it targets two different epitopes. It was first studied in Chinese patient population, and currently I'm heading up the phase 1 study in the United States with Janssen. So this particular experience shared the same interesting phenomenon, 100% response rates. The cell dose with this particular product, however, is much lower. It's almost a log lower than what you are giving for the Bluebird product. So the likelihood of CRS neurotoxicity appears to be lower with this product. So we're examining both of these products in expansion studies, in phase 2 studies with the Bluebird, and with a phase 1-2 study with this particular product. And you'll have more data that you're going to be hearing about this approach in another year or so. This is a summary of some of the other constructs with BCMA CAR Ts that have been presented and published on, but the main two that are really moving forward from a commercial standpoint are the ones in the two middle columns, the BB2121 and the LCAR product. The Novartis UPenn product has been, even though it was the first product that was presented at ASH almost 3 years ago, but it appears to be the most toxic one, and I don't think it's going to be moving forward commercially. So CAR Ts are not, they're not without side effects. These side effects appear to be short-term, however, and most are reversible. So because of, the cytokine release syndrome is kind of like a patient going into septic shock. It needs attention right away. There are a lot of different cytokines that mediate this, but the only cytokine that we have any control on is IL-6, because we have two different products that we can give to patients. And that's what we give, if patients do develop hypotension that's requiring, or hypoxemia that's requiring additional supportive care. So when we say grade 2 CRS, it's not a trivial matter. Patients are starting to develop hypoxemia. They're not responding to fluids when they're becoming hypotensive, so they might be developing, the need for pressor kind of regimens. The neurotoxicity does not correlate with cytokine release syndrome, so those two have to be looked at independently. Their timelines are a little different. Interestingly, however, with the BCMA CAR Ts, we're not seeing the same degree of cytokine release syndrome or neurotoxicity as we were with the CD19 product. Now, moving beyond the checkpoint inhibitors and the CAR Ts, there are a lot of monoclonal antibody-based treatments that are being developed for myeloma. The usual mechanisms of action, such as antibody-dependent cellular cytotoxicity, complement-dependent cytotoxicity, and direct apoptosis, have already been utilized by many different monoclonal antibodies. But what we're now seeing more is the two mechanisms of actions on the right side. You're going to be hearing a lot about them. So one is the antibody delivery of toxic payloads, so antibody drug conjugates, where the antibody, one of the arms of the antibody of the heavy chain has a toxic payload that gets delivered inside the cell. There are different platforms with different kind of toxic moieties that are attached, and the delivery can be from 2 moieties to 8 moieties per antibody. There are antibody drug conjugates being developed against BCMA. There's a GSK product that I'm going to share data about in just one slide. The other very exciting one is the bispecific T-cell engager or bispecific monoclonal antibodies, where this could be the poor man's CAR T. One arm of the heavy chain latches on to CD3 on a T cell, and the other arm can can latch on to any surface target on the cancer cell. In the case of myeloma, there are bispecifics they are being tested right now for BCMA, SLAMF7, as well as CD38. So the GSK 916 product is the first BCMA-directed antibody drug conjugate that has actually shown activity in multiple myeloma. And at the effective dose of 3.4 milligrams per kilogram, even though the data reported here is on 9 patients, you will see more updated data at ASH showing that the response rate of 60% is sustained. These are patients who are, again, pentorefractory and responding to a BCMA antibody drug conjugate. So this program is moving forward. There are some concerns about corneal toxicity with this particular ADC. The way that those side effects are being handled are by alternating the scheduled dosing as well as dose interruptions. And it doesn't appear like the dose interruptions are actually resulting in loss of response so far. The bispecific monoclonal antibodies are quite historic. They're not new. The concept was, I've shared with you that the holy grail of cancer medicine has been how to modulate the immune system. So the first concept was described in the 1960s, and the first human clinical trials were started in 1990 with GBM. But there was concern about cytokine release syndrome, and those studies were held. The same thing happened with the first CD19 bispecific antibody that was tried in 1995. Then the CD30 NK cell activating study that was started in 1997. And each of those studies were halted because of the concerns about cytokine release syndrome. And blinatumomab, that is now currently FDA approved for pediatric ALL, and there are clinical trials ongoing for other indications. It was first attempted in clinical trials back in 2001. And again, the studies were held because of cytokine release syndrome, and the patients, that particular patient passed away. Another company took up the drug and restarted in a very cautious mode, eventually leading to the approval of blinatumomab after a compassionate use protocol treating heavily pretreated pediatric ALL patients. So it got FDA and EMA approval. But the journey from bispecific monoclonal antibodies, back from being described in the 1960s to finally the first product being approved was almost 60 years. But right now, there are over 100 different bispecific constructs being examined in clinical trials. So you're going to hear a lot of data about bispecific monoclonal antibodies. As I described, for myeloma specifically, BCMA, CD38, and SLAMF7 are being examined. You will hear about an Amgen product at ASH if you're coming to San Diego in December. The data look very exciting. And I think the bispecific monoclonal antibodies, because they're off-the-shelf product, and CAR Ts actually require a lot of production, you will see that the bispecific monoclonal antibodies are probably going to have more clinical applicability. What about other targets? There are a lot of different targets on this slide. When I first presented this slide almost a decade ago in a conference, there were only 2 targets on it. And I'm probably missing a few more. But there are a lot of preclinical work being done with some of the other novel targets on this slide. And many of these products are already in the clinic, being developed. So what are the future directions? I think we have to incorporate the right IO strategies for the right patients. Not every patient can be approached with a curative intent. Yes, I'm talking about curative intent in the year 2018. I think we're getting to a place where we have so many different mechanisms of actions and IO that there will be subsets of myeloma that we would be able to cure. The older transplant-ineligible frail patients, we would be able to control the disease for a longer time. CAR Ts may not be for everyone. We have to design frontline trials, especially for high-risk patients. So there are 3 different clinical trials that will be starting in 2019, focused on high-risk patients. CAR Ts, we're just scratching the surface. Right now, we're talking about just BCMA, but there will be dual CAR T strategies. So CAR Ts developed that target 2 different surface markers on cancer cells, making this more cancer-specific. And there are certain disruptive technologies that will be able to help in decentralizing the production process. There are CRISPR technologies. There are desktop CAR Ts where CAR T technology is being developed where the technology will become cheaper. The bispecific monoclonal antibodies are going to be utilized more and more for patients who are unable to get CAR Ts. And then checkpoint inhibition, we have to figure out where they best work. And I think the post-transplantation setting and the late relapse setting with a non-mAb-based therapies is where they're going to be evaluated. I always like to acknowledge the patients and caregivers for participating in clinical trials. Wherever I go, I always acknowledge my mentor, Dr. Barlogi, without whom I wouldn't be where I am in my career. I thank my clinical team, my lab research team, as well as funding resources. And this last slide, even though I'm going to end with questions, I want to share this. This is a trip that I took with 8 myeloma patients, 7 caregivers, to raise money for Myeloma Research Foundation. This is the Mount Everest Base Camp. And it's a very arduous trip. Just to get to the base camp, it takes about 2 weeks. The fact that you can see 8 myeloma patients standing at the Everest Base Camp trek at 17,500 feet after having trekked over, Himalayas for over 10 days tells you that there's a lot of hope for myeloma patients in the year 2018. So I want to end with that, and I'll be happy to take any questions.
Thank you.